in very detail, or superficial about the therapeutic strategy in cancer research, how to design. You probably, have seen from last 2 days the many technologies that we have presented, and the in vitro assays, not so much in vitro, but in the today's presentation morning, I think you've seen a lot of in vitro studies. I think important is that many in vitro technology, I can say many technology, but might not work in each and every experiment, or the ideas that you're carrying to conduct your research, right? Sometimes you use this in vitro assay, sometimes you may not use that assay, sometimes you use something else, okay? So they were asking yesterday that what do we do with the drug when you start the work, right? You do the proliferation, invasion, metastasis, not metastasis, migration, colony formation assay. You can take the 3D cells and also culture, 2D cells you can culture, and do a lot of things. These are kind of a smaller, invasive, all these things. It's important experiments, MTT assay, viability assay. There are many assays you can do. These are all very important. And also in each one, you get some information that is useful to a lot of these things, right? So, but those are important, not enough, right? Not enough means that if you want to develop something, if you want to do something greater, probably those assays not enough. Probably you can publish a small paper and showing that this drug does this, and this is a natural compound, this is something you're working in your laboratory, or or you want to do something in a PhD program. It's enough. But if you want to do more, you have to bring the technology. You have to design your experiment differently, right? For example, I showed in the morning many of those in vitro assays she has conducted, right? Probably she might have conducted even more. I only showed you some of them. She might have put all the data in supplementary. If you go see the paper, probably you see more in vitro assays that she has done supplementary. But even though she has done many of the in vitro assays just in the main paper too. So, while designing therapeutic strategies, you have to know the technology. If you don't know, if you have not conducted, you should know your colleagues or someone else in the laboratory or someone else in the other lab, somewhere in the other university, and carrying or learning from them also important, right? Talking to them and also knowing what other experiment are, how they are conducting, what they are conducting, even the other laboratory. For example, you are from chemistry, you have to talk to a botanist or the zoologist or immunologist or some other biotechnologist, because some of the technique that they use, and probably it may be very helpful to you, right? That's, I think, key important. So what we're going to do now in this session, we try to understand the rationale behind the cancer design, like how you design your therapeutic cancer therapeutic strategies, and explore some of these methodologies. Already you know all the methodologies. I presented many things. You know the already concept, the tumor intrinsic, tumor extrinsic, all the work and the tumor microenvironment, kind of you touched everything. But here in this one, we just do the same thing in another way, another parallelly, something very different. Also, we'll discuss the translational clinical challenges that we're going to face, or the trials that we ran. So the example that I have given trials, for example, clinical trials, is for the phase 1 trial, right? There is a phase 2, there is a phase 3, and FDA approval of the drug. It's a big $1 million question, because they spend a lot of money. But from the phase 1 to phase 2, phase 3 is a lot of money, right? When from phase 2 to phase 3 is again a lot of money. But that investment, I and you will not be able to do it, okay? Someone else from the company or from the university maybe make a company and do it, or someone else will do it. But probably we will end up in phase 1 trial most of the time, right? But if you join a company, but you will see how the phase 2, phase 3 trials will be conducted. Probably they will be conducting, and you will know what are the parameters that you have to do and things like that. We're going to touch a little bit of those. And also important is molecular basis of cancer. You have to know the preclinical models. You have to know the molecular basis of a cancer, which cancer you are studying, and also the molecular infrastructure is different in each cancer. You all know already. So I have touched the pancreatic cancer, and the same way I have also explained the other cancer. What are the differences, like a mutations or any other regulator? For example, I was telling the other girl that when you take the calves or the fibroblasts, so in pancreatic cancer, fibroblast cells look the same like other cancer fibroblasts too, but those are the structural proteins. But if you go inside, the signaling probably will be different. The receptors on the top probably different, and the target may be different. And inside the genes regulate. For example, the Samara isolated all these calves, and I showed you. She isolated individual, all this population of the calves, and then she did all this sequencing of those. What are the genes are there inside and all. Those genes, for example, if you take out these calves or the fibroblasts from other cancer, like for example lung cancer, probably those signaling pathways may be different than this one, but maybe structural proteins are same. That's why the structure of the fibroblasts will be same. So these kind of things, the molecular basis of each cancer is different. In each cancer, the population of any of this micro environment, any of this cell population is different. So you need to understand that before you carry preclinical models. When it comes to preclinical model, see, for example, I use the KRAS mutation, P53 mutation. In lung, they may use it differently because the mutation in the RAS is only the 40 to 50% in lung, and there are other mutations maybe. So you use the model according to the mutation. And what is that important? Initiation of the cancer. In each cancer, the mutation profile is different. So, but the technology is same. The way that we conducted in the pancreas is the same way you conduct in other cancers too. So what I'm saying is that preclinical models they use to study in different cancer is different. They have different models, but study the same way. Okay, you have to isolate all these populations, study the tumor intrinsic. You have to see the molecular level, cellular signaling, and all those. So when it comes to combination therapy, combination strategies that we did in the pancreatic cancer, you saw all those how I did yesterday and today, and the same way you design in other cancers too. But in some cancer, probably targeting a single drug may be okay. Okay, because they're all not tough tumors. Okay, and some cancers you may have to do the combination therapy, and with the chemotherapy, with the immunotherapy, like that. So in melanoma, you have to do the immunotherapy because it's very important to do the immunotherapy like that. So you have to know, like, what are those, bases, that strategies that when you take different cancer. Precision medicine. So this is like huge thing, right? Nowadays, everybody is talking about precision medicine. They are talking, but important is that whether how you conduct precision medicine, or you have to collaborate with someone, companies, or someone who is actually doing those kind of methodologies in their laboratory. So we touched a little bit of this molecular basis of the cancer. Overview of oncogenic pathway means that in some cancer EGFR mutation is important. In some cancer P53, some cancer KRAS. So when they analyze this in this paper, the Cell paper, the 10 cancer pathways. There are 10 different cancer pathways. In each cancer, different pathway, but they're all pathways though. Okay, and also there is when they analyze all these things, they got into like brain has like 628 some in that different mutation, different copy number, methylation expression. Though in each cancer, if you take a leg, something cancer here, so they have a different signaling and different things. Okay, that's I think important to you to know that they are different. And also when they did the drug study, for example, 100 plus cancer drug studies, and where they saw how this combination work, how the synergy of this combination work. Okay, all single drug, how that is going to work. So this information is very important for you. Okay, there is tons of data out there. This is 2018. Remember, 2018 to today is almost 7 years. Just imagine over this one, what and all the people published. Okay, amazingly, there are tons of data out there that people publish by using this type of technology. They bring another technology ideas. So you have to have those literature with you, understanding it. If you cannot understand, try best to talk to someone or communicate to someone and try to dig more and more. Don't leave it, because maybe you're losing something very important thing in that paper, right? It's very important. And also we talked about genetic and epigenetic alterations, and these are all the same thing, which we discussed during last 2 days. Driving the tumorigenesis means that, again I'm coming back to RAS and 53NE mutation. Tumor heterogeneity is and its implications. So now you saw our own study yesterday and today, when we talked about tumor intrinsic targeting these drugs are not enough. Then we went on and did the immunotherapy. So means that you have to understand the tumor heterogeneity. If you don't understand those heterogeneity, like we were not able to, because there was no technology to understand. Now today you can understand it. You can isolate those cells and do a lot of different things, and or someone might have already published, understand from the data and see what this population is doing and where the genes and which one is important. Okay, if we can understand, in our own study we were able to understand, and then our survival was boom, like unimaginable survival we got. So the same way you're missing something. That's why you need to understand the tumor heterogeneity. That's why people are saying, please go understand heterogeneity. Then you can implicate your cancer or the therapy better, right? So validating the therapeutic targets, right? So how do you validate the therapeutic targets? For example, somebody asked me yesterday that a different concentration of the drug, or how do you actually think how much to use in vivo, how much to use in vitro, these kind of things. One thing that we used to do, we used to run, because I had a MD chief that also worked with MDs, so they knew clinically how much they are using the drug. For example, dasatinib, that's a SRC inhibitor. My mentor, we knew how much they are using in this clinically, in a SRC inhibitor, because it was clinically approved. Remember, I always said, you use the clinically approved drugs if you can. If there is a new target you found out, probably there is no inhibitor, it's okay, but you are actually finding it. Probably you end up publishing it, not going forward, because that is not clinically approved. If you have to get the approval or doing something, you have to be attached with a clinician or medical oncologist and conduct those kind of experiments. There are a lot of companies in India actually just do their clinical trials, right? There are many companies. You can actually talk to them and see whether what they are doing, what drug they are using in clinically, and you can actually attach to them, combine. They will love to have the collaboration from academia. Only thing is you have to go and attach to them, because you want to understand what they are using, and they don't want to be with you because you're still doing the basic science like 10, 15 years ago. They are beyond, right? So you have to be in their level and to compare your study to them. Then they will come to you and they want to use your data and they want to combine and collaborate with you. So your science has to go to next level. Then only they can come, right? You have to develop those kind of, for example, you have to know what is orthotopic model, what is xenograft, what is PDXs, what are those organoids, what are these spheroids, how do you isolate organoids, how do you make the spheroids. If you don't have those kind of knowledge, if someone is talking from the company, probably you will not be able to collaborate with them. So you have to develop those knowledge, and also once you develop the knowledge, go see if someone is conducting this kind of experiments. Nobody says no to you, right? Say there may be other university, there may be other, like you all came to this course, you're learning something. For example, if you would have not, probably you would have lost something, like not able to, right? You're maybe a cancer biologist and you're maybe you're isolated in somewhere and doing, probably not getting exposure of something, these technologies and methodologies. Probably this is one platform that probably you're getting ideas from someone who are really doing their cancer biology from last 25 years. It's important. If someone is giving you a hint, yes, even though it is a small thing, doesn't matter where, you have to take a bus and go go meet them and see what they are telling. Probably there is always something gives you an idea. For example, in Vanderbilt, I think there was a Nobel laureate. I always, when they have a Nobel lectures, I just go, because it is not for what experiment they have conducted, it is the experience what they went through to conduct those experiments, right? For example, there's someone is coming a Nobel laureate somewhere, right? It's not that you're going to see the Nobel laureate, it's about the experience, what they went through to conduct those kind of experiments. And that actually inspires you to do something better in your life, right? It's not that Nobel laureate, like there may be someone who is pioneer in one work, pioneer in some research, a chemist or someone developed the drug who are actually giving you a talk. Probably there are some inspirational talks. Plus, more than that, there are some technology-oriented investigators. They give you a technological talks, which actually might help you. But you might be not doing the technology, but you're getting the idea of those technologies itself is actually a huge thing for your research, right? You may be like, working in the laboratory for so long, but you will not be able to, understand what's going on. But sometime when you go out like this, probably you get an idea. I think that idea might change a lot of things. That happened to us. That happened to us many, many, many times. So I think it's a key. For example, someone is presenting from the pediatric cancer. I have attended pediatric cancer, but my lab people says there's a pediatric cancer, I don't want to go. I know what they are thinking, but my thinking is different. But if I go there and see, maybe probably one day I will understand what is pediatric cancer is. So what is that they are actually going through? What these actually kids are having a issue? Why these kids are getting cancer early on? So that itself, someone is presenting who is experienced and who knows exactly what's going on, and that experience that actually you're getting that information for yourself, understanding that biology probably help you how to do it in your. For example, the cancer, pancreatic cancer. So it's pancreatic cancer kind of similar to a prostate cancer, right? Probably you might not know, but I just understood from one another talk, who is expert in pancreatic cancer is called David Davidson. So he was actually talking about pancreatic cancer biology probably related to prostate cancer. I started digging all this prostate cancer biology. I went to these talks in prostate cancer biology because that is actually giving me a hint how to study my biology, my cancer, right? Some hint. Because each cancer is different, but some cancers are all related. Means the way that they develop into cancer, right? Some of these high throughput screening omics technologies are important. Okay, when it also depends on kind of drug study or why you want to do the high throughput screening and what is your intention. Intention is to investigate something new, and yes, you have to do the high throughput screening. Your question was something related to developing some small science. You don't need a high throughput screening. All you have to do is dig and something literature survey and collect all this information and do it, right? It's all about what you want to do, right? And using the informatics and genomic databases, for example, TCGA have written, but there are a lot of bioinformatics tools available nowadays, right? There was a student I talked to, she's from Bangalore, and she was doing MS program in Boston University. When I was talking to her, I was amazed to know, she's from Bangalore, but what amazed to know, so I just got in contact. She was actually searching for internship. And I asked her, what are you doing? She's exactly doing what I wanted to, someone bioinformatician to do. I was like amazed to see, sometimes you never know, you cross with someone and that person is actually doing something that exactly what maybe you may be doing, right? It's just happened to us and I want to hire her. It is like that. So we're going to hire her. So because that is exactly what we wanted, particularly right now what we are going through, right? Similarly, sometimes what happens, what you cross, and when you're talking to another person, nowadays phone is an issue, right? So we will be on the phone, we will not be able to talk to anyone. Sometimes I think better to talk to something, better to leave the phone and talking to something. Probably you get a lot of information. Sometimes you have many questions will be solved, right? It's just a human nature. The technology you use for validating a lot of assays. For example, you use cell viability, apoptosis, proliferation. These are all in vitro, in vivo xenograft, genetically engineered mouse models. Like that, many models are available. I don't have to list out all of them, but important, I think, the concept and ideas that you develop to use those models, how to use. For example, you may not be able to use it here, right? Someone is already doing it. You can collaborate with them. But your science has to go to that level, probably to collaborate also. But your science the in vitro assays and in vitro studies has to go to that level. Then only that person will come and tell you, because that person is actually working on the in vivo, and he will not be able to work on the in vitro, because there is no knowledge. There is no person, there's a labor work, like a lot of work in vitro. Like actually more than in vivo, I think you work in vitro a lot, because there are a lot of variability. Today you go to cell culture and you conduct the cells and all those things, and then you go after a week, probably your data will be different. Even the same cells, same culture room, it can happen. There are a lot of variability are there in vitro cells, and then you conduct one of the qPCR, and then after some time, probably you may not get the same fold change sometime, right? The variabilities are there. It's very common, but important, I think, validating those parameters, right? When it comes to a preclinical model system, right? During the drug development, you use the cell culture, which you all doing probably, and there is a 3D organoids, because if you want to study the microenvironment, if you want to study how this drug is transferring through the membranes and how they, cell attachment on all these microenvironment, you have to create a microenvironment to the cells. You have to use the 3D organoids. 2D is not going to be enough for you, because your 2D, you're not going to get all those information. It all depends on, again, your scientific question in your study, okay? That study question should take you there, okay? And in vivo models, all these models we talked about. So when you're using in vivo model, xenograft, so why you use xenograft? Probably if you want to just to know what happens to the tumor weight, tumor volume, that's all you get the information. You cannot take the xenograft tissue and study all this immune other population. No, you're not going to get the immune population. They are under the skin. You're not going to get all those population, because the tumors are not in the right spot. You have to have those tumors in the pancreas or the lung or the lung or in brain or wherever that's supposed to be. Then only you can study the tumor microenvironment, right? You're studying the wrong model. Many people published in a xenograft and then take the immune cells, and I've seen many papers. But if that comes to a review in my table, probably I will reject those papers, right? So it's exactly because that is not a right model. So like that, you need to know what these technologies do, but that you have to develop a knowledge. That knowledge is just from the publication paper is enough. You don't have to talk to someone. It is available already. You need to know how to search and get those information. Searching is a key factor. Searching itself is 50% of your research is done. All you have to do is another 50%. If you don't know search, that searching, right, in the literature searching and right way, okay, it will be very difficult to do research, good research, right? You need to develop that searching, and you learn from yourself though. There's not someone is not going to teach you how to search all these things and get. You fail and you search again, and you learn something, and again you search, right? There are some basics you can get it, but what I'm saying is, at the end of the day, you develop your searching knowledge yourself, that how to do better, okay? And there are challenges, there is no doubt. The reproducibility issue in science, right? In your own biology or technology, there is a reproducibility issue. Everyone is facing, right? So it is very common. But the way I have explained to you last 2 days, like we don't have the reproducibility issue. That is the reason that they went to a clinical trial. There is no way we have an issue in the reproducibility. The reason I will tell you, it is the way that you model your study, the way we validate every step. There's just not one or two, there are 3 or 4 times you have to modulate your study, right? For example, if it is not a KPC mice and the PKT mice, you take another mice and probably study, probably there will be a problem or maybe a issue. But the model that I have showed you and studied, those models will not have a reproducibility issue, because that has been conducted in many times, conducted from different ways, making sure that is actually working. So it's, I think, important as a scientist that you have to develop the study, a reproducible study. I think that's a key factor, right? There are these things so that each one, I'm just saying, this is a PDX model here, is like testing, how to test, and all those things. But it's actually a nice paper here. It's actually very recent, 2023. I loved this paper, so you just actually see what you can do. When it comes to drug discovery and screening approaches, right? There are a lot of people high throughput screening they've done. So for a small molecule library, which I'm going to explain to you tomorrow, when we do the natural compound screening and stuff. And there is a new technology, RNAi screening, CRISPR screen. These are all new things. Believe me, there's like fantastic data. I think it's all about what you want to do, how what's your research, okay? What's your plan? It's all about that. So I did not show any of those screenings so far, but I'm going to show you a little bit tomorrow. But the important, I think, why you want to screen, why I screen those compounds, I'll tell you that story tomorrow. But I think your science has to take you. So your problem or the questions that you have in your biology should take you whether you should screen or no, and what for, and what questions. So we'll discuss that tomorrow. So another optimizing the preclinical mice model or the preclinical studies. I showed you today morning that how did we optimize? We optimized using different model system, but they're all the same. They progress and become a cancer. And also we optimized with the orthotopic model and the xenograft model and PDX model. We want to compare with the human tissue and compare that data. And also there are a lot of organoid models that I did not show, but there are a lot of organoid models we have developed, spheroid models we have developed. So you have to show that indifferently optimize all those studies and making sure that preclinically you are stronger. The one another thing is when you are doing the study like all this chemotherapy, what I did not show you is that pharmacokinetics. How did we do the pharmacokinetics? And why we did the pharmacokinetics? How did we analyze the toxicity and the measurement of the toxicity? And what parameters did we use to measure the toxicity? Those kind of things. These are all done by our a team of pharmacologists and a team of pharmacokinetics people, a different group. Because remember, your expertise are different. Our expertise are not pharmacokinetics analysis or toxicity studies. Only we understand and understand those biology and things. But they are the experts. They just run those experiments and regularly. I think there are a lot of, in India, there are a lot of pharmacokinetic studies conducted outside small companies, outskirts in Bangalore. I've seen many. So it's like that. You can actually, you have to get some money and go there and conduct those pharmacokinetics because they're experts. They do. Once you know how they have done, probably you can manage to do yourself or with someone, someone colleague or other university, someone collaborator. So I think it's important to use the expertise when you are doing the preclinical development, especially pharmacokinetic analysis, toxicity measurement, right? When it comes to a combination therapy, when you want to add drugs together, right, and overcoming therapeutic resistance. Say for example, I showed you example SRC EGFR inhibitor. How did I do it? On MEK STAT3 inhibitor, how did I do it with adding the immunotherapy or immune checkpoint? These are some of the examples just I showed you. But that was conducted 10 years ago. Those experiments were conducted 10 years ago or 7, 8 years ago. But remember, today what we are doing is entirely different. The combination therapy and merging together two drugs, we have a lot of technology today. Okay, we didn't have that at that time. So the same way like how I'm talking about single cell sequencing, single population, different population, the same way when it comes to a drug study, there is a new technology, synergy study. And you don't have to worry so much like before. We don't have to go wrong. There is technology developed. So you can actually use those synergy development and also overcoming resistance and all those things. So I'm gonna, I think, hit that in the next 2 slides, and I can show you a little bit maybe there. So, and also when we are doing the preclinical development and the post-clinical, first after phase 1, right? You have to do the, biomarker analysis, making sure that the study, whatever you are doing, are conducted in the preclinical or the post-clinical. I was talking about the pharmacokinetics analysis and the toxicity analysis. You have to do those, monitor those, analysis, and I'm not going in detail of the what analysis you have to do. I think you can actually get it, in literature survey. And important, I think you have to do those toxicity analysis is either in the preclinical model or in the post-clinical or after the phase 1, after the patient data that you collect. Remember, if you are collaborating with a medical oncologist and if they are ready to take your drug to study in their clinic, okay, believe me, you get all those data because your study, they are actually doing it because they believed in your work. But they will give you the data. They will give you the blood samples, tissue sample, everything. You can do a lot of toxicity studies, a lot of different biomarker study that actually helps you to develop a phase 2 trial, okay? So when it comes to preclinical or translation research, and we always talked about the phase 1, right? So, and there is a phase 2, there is a phase 3. It's, as I said already, we don't do those phase 2 and phase 3 in my own because I'm a researcher. I'll end up in phase 1, and then if that is working, that phase 2 will be conducted completely by the clinicians and the medical oncologists completely. That is not my work. But what I'm saying is that taking into phase 1, making sure that phase 1 works is a key important point as in, biologists, cancer biologists, therapists, or preclinical developmental. Like, you have to know all those parameters before it goes to the next level, because there are a lot of patient involved, a lot of other things are, analyzed and differently than that what we do. So your drug or your study should go to phase 1. That is, I think, key important point that you should know. There may be a patient sample, like maximum is 15. They may conduct, maybe it will go to 20 patients, that's all. But when you go to phase 2, there will be more patients. And phase 3 is tons of patients, right? They were going to do the multi-institutional, multi-hospital studies and all those things, right? So those challenges are entirely different than what challenges as a biologist we do and conduct in the laboratory. So when you try to design clinical trial, right? That is not our work, but I'm trying to explain to you that when we designed to the phase 1, we did not design. Our science took us there, right? Our science, but when you are designing the therapeutic experiment, for example, Samara designed the AIM-3 with the therapeutic intention of doing, but probably it may go, it may not go because of survival issue. But when she is designing the AIM-3, but when you are submitting the paper to a high impact paper journal, probably they're going to ask you, because they want to translate the study. They want to show, you have to show the translational relevance to your study. They're going to ask you whether, did you do the chemotherapy? Did you do the immunotherapy with this, blah, 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 blah. Also, but you have to know how to do those things and so to develop a trial for them, right? Trial in the future. So phase 1 trial objective is the safety, dosing, pharmacodynamics. That's all. So that's all they collect the information. And designing those biomarker guided enrollment and all those things, it is the next stage. But after the trial 1, even though in the trial 1 they actually do it a little bit, okay? So the recent trials combining, like, what we did, examples I already showed you yesterday and today, not today, yesterday. I showed you all those 2 trials, a phase 1 trial, and do you know all the story why we added the immunotherapy, right? You know the story. You know the story, without immunotherapy, we also went for a phase 1 trial. You know that story too. So that story is enough for you to design your kind of experiment you want to do. Okay, that actually a good story for you. So there is no need to go what exactly clinical trial will be conducted, because we are not clinicians to conduct the trial or how they do and what they do and understanding it. Okay. So the successful combination therapy, for example, I was telling you about, okay, this is a Nature paper you should read. This is a very recent paper in 2022, because here they're going to explain to you how to do the drug combination, okay. They have done 2025 drug combination study, And they screened in breast cancer, lung cancer, and pancreas cancer cell lines. Like, there are like 125 samples. They also studied the synergy. They evaluated on the basis of efficacy and the potency. I think almost like 108,000 drug responses. Okay, and also they have they have identified the biomarkers. They have like in here you see the heat map where they see they show you the IC50 values for 1,275 combinations. They combined 51 breast cancer cell line. They have clusters. They're actually clustering and all this. There are a lot of huge information here. I'm telling you, these combinations are available in this paper. Okay, that's what I'm telling you. This is 22. You were in 25. Just imagine in these 3 years, how many other papers came out, and how many drugs or combinations are studied. You don't have to go anywhere and search for it for the science. You can actually get by sitting in here. You don't without a test tube. You can actually do it if you have a knowledge idea how to use this kind of data and analyze and think in your own model. Analyze and think in your own lab, somewhere sitting at home or laboratory. Okay, believe me, you can actually get it. But it's about your awareness. Yesterday I completely told you, awareness. You have to develop that. Read papers, understand it. There are a lot of literature you have to read, day and night. Get up and start reading and search for new paper every day. So this is the key thing. So that's why otherwise our knowledge will be like we will not be knowing all these things. What's going on? It's already what you're working on. Probably it's already done like 15 years ago. Probably you're just running because you want to run the lab, or you want to just do the PhD, but you're just doing it because the sake of doing the PhD. So you may be missing a lot of information that you needed those information. I think that you should not miss it. You have to. For example, people don't. I know many of them. They don't go and read the Nature paper because you don't understand the Nature language. Okay, and believe me, they don't understand the Nature language. Take the dictionary and read about it. I'm telling you, you will learn a lot from those papers. But nature is nature. Okay, that's why you will not be able to understand. But it's okay. Okay, but read, understand, and otherwise talk to someone and discuss. Okay, you can actually get it. These are all English only, but it's about the language they use. Sometimes probably it is not common words that you're going to use it. You use it differently. You have to develop those skills too. And probably if you're not an English speaker, I was not. I studied in Kannada language up to 10th grade, and I went to PUC and I just started using the dictionary. So that's how I learned a little bit of English at that time. So what I'm saying is that it's important you learn yourself if you want to live in the world, in scientific world. You have to learn English, right? And especially the words they use differently, you have to use it. I think this is the great paper. I think you should do it. And this is the same figure I have taken from the same paper in the Nature paper. Basically, what they are trying to establish here is the synergy between many of these cancers I'm talking about. There are a lot of drugs that already know. You don't have to search for it. I'm telling you, there is synergy. They already know. If you see this figure, you already know. There are a lot of drugs, approved drugs, synergy. They can tell you already what exactly is going on. You don't have to search for it. That is the awareness I'm telling you. There are a lot of studies has been conducted already before you even think about it. Okay, so that's why this kind of papers, there are a lot of tons of papers are out there. Collect those information, collect those studies. One paper is enough for you. One paper you can get another one within yourself. Okay, because the knowledge, how you are thinking in your laboratory is different than what I am thinking in my laboratory. Every human mind is different. The way you think, maybe next person will not be thinking. Okay, your thinking may be different. That means that there is a better or worse, that's secondary. But thinking is always different. Okay, when you read one paper, I'll tell you, 10 people think differently about that paper. But there is a common factor. One is that surviving it, or whatever the conclusion is the same thing. But your thinking is different. Means that there is a 10 paper, but the conclusion is the same thing. But the way you conduct the experiment, 10 people, is entirely different. Okay? So what I'm trying to tell you is that the data is out there. Okay? You may have to start thinking and searching information. Okay? And important, I think, another key lesson here is that integrating the therapeutic approach means that you may be doing, for example, in another laboratory, someone else is doing some other type of therapeutic, probably same drug. Okay, for example, MEK inhibitor, right? In some other laboratory, another person published another MEK inhibitor and published differently. And you have to collect that information. Like trametinib is one drug, another one is MEK inhibitor, another drug from different company, AstraZeneca company, some other company. So there may be tons of paper. You have to collect all this paper. Take the common factor from all this paper. Believe me, you will be amazed to see what's going on here. You'll get a new paper, your own, when you collect all this information. That's the beauty of these drugs, right? Each drug act differently, do differently, even though their target may be the same. Okay. There are a lot of immunotherapy combinations are coming out. Okay, nanotechnology, AI-driven drug discovery. There are a lot of AI-driven drug discovery because it's easy to get the information worldwide and collect. When you type something, you actually get that information, whether it is right or not. That is you have to analyze and you have to dig those information, and you as a scientist, you have to make sure that that's the right way of getting the data out. But information is out there. Okay, there is technology developed. Okay, model has been developed. Nanotechnology also another way. But in nanotechnology, when it comes to nanotechnology, you need to know that why you want to do the nanotechnology. Not because someone else is doing. Not because it's a pioneer, not because if the technology is a great technology. No, it's about your scientific problem. For your scientific problem, you have to develop the nanotechnology. Okay, what is your scientific problem? You have to first find out before you do the nanotechnology. Okay, for example, you want to target MDSCs. Okay, you have to target the macrophages, and you don't have the drug directly to target the macrophages. And you want to do the nanotechnology, specifically bind some factor that will go only to the macrophages and hit the macrophages, and then you can actually kill the macrophages and do something activating some signaling there in the macrophages, then you study. So if your problem, scientific problem, is macrophage related, that's what I'm trying to tell you. So your science has to take you to nanotechnology. Nanotechnology now should not come to you. Okay? So you have to bring the problem first, and then think about what technology you want to use it. Okay? So immunotherapy combination, I already told you example, our own example, why we did the immunotherapy. Again and again, as I'm telling you, it's the same thing though. It doesn't matter which cancer you're going to take, you end up doing the immunotherapy. That's why this common factor I keep telling, it's a common. It doesn't matter which you're going to work on, you're going to do the immunotherapy. So if you cannot do the immunotherapy, it's okay. It's about your work, where it is going is a key factor. Don't worry about immunotherapy if you don't want to do it, or if your paper is not impactful. It's okay if you want to just do the normal thing, it's okay. But do it right. Do it right, validate right, and make sure that is actually reproducible. I think that's the key. Otherwise, you'll not able to sleep later on. Okay. Overall, I think recapping all this work is that integrating approach, molecular mechanism to a clinical trial. So combination, biomarker study. If you think the translational approach is essential for your therapy, but you have to really, the way that you conduct the experiment from the beginning has to be different. Okay? If you want to take your study to a translational, I'm telling you from last 2 days, the way that we have approached, because we were not approaching our science to a translation. We did not know when we did the first experiment, we're going to do the translational. We did not know. But it just took us there. But later, we planned such a way that if we are going to do the translational, and we failed also in one of the experiments, but after that, we went back and figured it out. But important, I think, is that even though we failed, we published in a good paper, but it's not enough, right? The way you start your experiment, for thinking what you want to do in the later on, I think the key factor, right? And your impact factor is 5 or 10 or 20 or 23 or 25 or 50. You have to decide how much work you want to do. Okay, 5, how much work you need, and 50, how much work you need. You have to think differently. Okay, how much collaboration you have to do. Okay, if you want to do collaborative research, then the impact is going to be higher. So that means that you have to go through all these problems and figure it out together, right? So like that. It's about what you want to do, right? How you want to do, where you want to do, and where you want to go from this paper or the work, right? It's about that. So that's the story. Just gave you a wrap up of all the work that we are actually conducting, and also discussed last 2 days. I think if you have any questions, I would like to answer. Any questions?
Hello.
Any questions from the morning? Any questions from today, any day, any talks? Anybody has any doubt on anything? So, who is actually conducting experiment right now in the laboratory? Who is actually doing some experiments in the laboratory? No one? Are all scared? Okay, so you're doing the PhD. So that's why you're doing the experiments. Okay. How many of you are doing PhD? So that's many. So that's good. So I didn't know that this many. I knew that there are many. But that means that definitely is helpful for you. So what type of research you do? I cannot ask you, is it cancer biology or something else? Who is doing cancer biology? Cancer related. Antioxidant is a plant extract study. So is it something natural compound? Some other than cancer, how many? Okay. That's good to know. So you're from food technology, you do food technology. Who else is food technology? Food related. Good. So see, even though you food technology, let me tell you, I was from food technology too. So I'm from food biochemistry. I did a PhD in DFRL. So when I started, I was actually screening the rations for army. Okay, rations, biochemical analysis of rations, and biochemical analysis of all this army, that Silcher and all those people, like freezing condition, non-freezing condition, regular temperature. What is the difference, and what the biochemical assay. We did publish a couple of papers during that time. So also I was running all the GC, mass spec analysis, HPLC analysis, of all those things. So for the food only, it's a biochemistry. Then my PhD was actually on the aging. That means the food aging, right? Meat aging, and because we want to increase the aging, we want to decrease the aging so that we can actually eat it even longer for the army purpose, right? It was important for the army. So I was actually studying to decrease the aging so that we can actually keep the meat for a long time. So, but I've done the biology of it. I studied the biology of it because I want to figure it out. How do I do that? For that, I needed science within. I can put the spices. I can increase, stability. I can do a lot of spices, Indian spices, and mix and match and all those things. And then I think that you can actually store it for a longer time. But that's what exactly we do at home, right? We mix with spices and marinate, and then much better. So, but we can do that. But important, I think, in biology, how do I reverse the aging? That was actually interesting to me. So that's what I did, and that is my PhD program. But we did finally a little bit of that. But what I'm saying. So, but I learned a lot in biology wise. But I learned how do I change it. But it may be probably helping some army, someone somewhere. But how they have used that technology somewhere, something for something, it's a great thing. But good relief, good feeling, right? Because it doesn't matter what research you do, but end of the day, if you do something interesting and conduct something interesting, which actually gives you values to something, was actually great feeling. But my interest was some of the factors I studied during my PhD program, during the food biochemistry analysis, whatever, and whenever I was doing PhD program, those factors, the enzymes and the degrades those actin, nebulin, and anything, all those structural proteins was like cathepsins and calpains and proteases and all those things. But those are important in cancer also, and during that time. That is the reason that I developed the cancer biology during that time. Means that I always wanted to do cancer biology somehow. But in India, I think at that time there was very less cancer biology, investigators around at that time. So I was actually searching for cancer biology. I went there to US to do the cancer biology. That's all, nothing else. So I joined the aging lab to initially to understand whatever I did, the aging related, that whatever the even the small protein, I wanted to know, because the lady that I joined, she's a space DARPA project she used to do for the defense project. It's actually about aging again. When they, astronauts go and come back, then she was analyzing all those blood samples and all those, biology, and seeing what happens to those factors and all. So, but for me, that was like an exciting factor, because I did the same thing in a smaller level, but I wanted to study and understand how she is actually conducting experiments. I went there and I did for 6 months or 7 months. Then I realized that that is not me, because that I cannot go beyond something in that field. Then I went back to cancer biology, because that is my passion. So when I went to cancer biology, I was like relieved completely, because it is a passion that you believe in something that you wanted to do always. So I went there. But still, when it comes to aging or aging studies and all this, I get excited, because even though I open some paper, and if anything like aging related, I always go back and look at it and see what's going on. That's what you do. So it doesn't matter what you do, but at the end of the day, that you always go back and look at it. And I think that's important. It doesn't matter what protein you're studying, whether you're doing about botany or zoology or genetics or food technology, food science, processing or not. The passion that you develop, right? The studies that you're conducting in your laboratory, it has to be something novel, has to be something new, has to be something that you are the one, the pioneer. Okay, you are the one that conducted and investigated. No one in the world has done that type of experiment. Even though you sit in that corner, doesn't matter where you sit, Davangere or Shivamogga or Mysore or Bangalore or Tumkur, whatever, wherever. Okay, wherever you sit there in that corner is important, that what you do, right? That's why this literature nowadays is so good that anybody can get any information anywhere. It was like, we were not able to get even the one paper to read. So I used to go to CFT, DFRL people, the defense laboratory people, can go to CFTRI and use the library. That's the advantage what we had. So I went to CFTRI every day in the evening after 5 o'clock or 6 o'clock, whenever, because the defense laboratory closes 5 o'clock. They only work from 9 to 5. When then the director changed, and my boss became a director, and then things have changed, because I can stay in the laboratory and I can work more because my boss was a director. But before that it was difficult, right? And then during that time, after 5, when they close it, I used to go to CFTRI and read all this related, because it's all food related only. So all these papers and everything was available in CFTRI at that time. So I used to go use the library every day. So I think it's important. You have to search for the information. One another story I want to tell, I always told Joshi. Joshi knows I did not do my experiment, a lot of experiments at DFRL at all. Okay, my experiments, I only was running the gel, and only the photographer Raju was taking the photographs, and I had to go and develop my photos in some other, in a studio in Mysore. I used to go to Gandhi Square, around that area. There's one, photo studio, and then I used to come back and I request Raju again every other week, and I was like, how difficult it is in the university. There's one photographer, and has to come and take a photo of your gel, and it's embarrassing to him, and it's like, for me it is life. So I don't know how to take a photo. There was no camera, there was nothing, that we don't know how to develop all those things during that time. And I used to, to run all these things. Remember, these are all structural proteins. So I had to run a gel, like big gel, and the proteins, there are tons of proteins. I don't know which protein is what. I had to find something new protein that no one has seen. That is what my intention is, because I was doing the aging work. So then another one, I used the microscope in zoology in Mysore, and I used to go and use the microscope because there was no microscopy in DFRL that I can use. And then I developed the mice, the antibody for actually isolated the nebulin protein from the gel and then crushed and isolated and all those. And I used to inject in the Mysore University biochemistry department to the rabbit because I was not able to use it in my DFRL. And inject a rabbit and take the blood and isolate all these protein and things like that. And then when I wanted to do the electron microscope, because it's a structural protein, I had to go to NIMANS, and there is a electron microscopy there. I used to go to NIMANS every other 2-3 months once, and I used to stay there, my uncle's house, and then I used to go there and conduct those experiments, because I had to collect the meat, I had to buy a meat and age them, and we put it in a different fridge, and all those things. And then take the samples to them and cut it and then do analysis and all those. And some of the experiments, I used the friends from IASC and laboratory there, and my things like that. There are many different university institutes I used to run for all those experiments. That's why it came out beautifully, right? When you get a poster award, first award, like oh my God, it's mine. I don't know, like unimaginable, because they see it. When they see it, the effort that you put in, you can actually show up, right? So it's, I think, important. I think the work has to take you, right? You have to have the interest. If you don't have that interest, it's very difficult for you to, long run, publish or do anything. Today, if you see why I was able to accomplish any of these things, because I have passion myself. It's only interest. It's not, my boss was not telling me, my mentor was not telling me to do this experiment. I used to conduct, and I used to go and show, this is what is the changes, this is the paper says this, and this is what I got, and I don't know what to do next, and I'm trying to establish. Then they will give, hints somewhere here and there, mentors, but they may not know the details of it, but the experience they'll give you a hint that somewhere this is how probably is working, right? So that itself is huge. So I think why I ask you different, this thing doesn't matter which one you're working on. You all can become a cancer biologist. You can all become something else. All become a food technology if you want to. If you all like food. So it's about that. It's about your passion. Okay, you can actually develop many investigators from cancer biology. They are from chemistry. They are from engineer department. There are many investigators. They are engineers. And I told you one of the example is the sonic, technology that I use for the collagen. She's completely engineer. She changed all the technology to a cancer biology now, and she is doing great. She has got a lot of grants and things like that. Because technology, if you learn something that you can adopt to some other biology, and you can use it. So, for example, if you learn nanotechnology, if you know how to do nanotechnology, believe me, there are a lot of investigators you can collaborate and do those things. You'll be a pioneer in one day, in a couple of days. Like that, there are my friend in Vemula, in Indian Institute of Science, is actually a chemist. He was in Harvard, and he came back to India, became faculty. So he actually designed one of the drugs that I'm going to tell you tomorrow. He designed the drug, natural drug, synthetic drug. Believe me, he is expert in chemistry. He has even companies in Boston, and is that expert. Means that even though you're sitting here, your company is running in Boston. Can you imagine? Like that. So if you're expert in something, believe me, you can actually do greater things, in your life. It's about, that's why I was telling you in my first talk that you had to be expert in something. You have to find something that you are interested in. Your mind says you are this, and your body says, no, don't do that, do this. You better do this, because probably you become expert one day, right? Probably that itself is more than enough. You don't have to do much, right? So So this is a question and answer discussion question. If you want to ask anything, please go ahead. We're going to do the one tutorial that is, I think, the targeting cellular signaling and tumor microenvironment. I think that's the morning session.
Yes, sir.
Yeah, go ahead.
Sir.
Yeah.
Yeah. Actually, you answer my question because I want to ask you why you don't go to the phase 2. Okay? But my question, you got a good result from phase 1 result. What you will do to convince the investors to proceed in the next level? As you say that because you are just researcher and you need a clinician, for example. What you will do to convince other investors or the funders to proceed in your result to your next level of phase 2?
So you're saying if you don't do the experiment in the test tube, like how do you convince your clinicians?
Yeah, I mean that you stop in the phase 1
Trial.
right. What you will do to go to convince the investors or the funders to go on the
Go to the phase 2, right?
Yeah.
About your toxicity, your drug combination has to have the less toxicity and also survival, increase in the survival in a cancer patient, right? You will have in the phase 1, you will have like 15 to 20 patients, right? So in the 15, 20 patients, you need to have a good statistics of these patients that these patients survived better than like how you actually showed in the published paper. Like, you survived better, right? At least you should show similar results or approximately that your patients are survived. For example, your patients were surviving before 5 months. At least if you can show like, these patients have survived like 6 months, 7 months, 8 months, 9 months. That is one factor. Another factor is toxicity. You have to have a less toxicity, right? We're going to talk a little bit about that tomorrow, a little bit about it, and then how that toxicity is important, like how to take care of the toxicity if that is something. If anybody is interested to work on, that's a great area too. So just the toxicity, you don't have to do anything. Take care of the toxicity, okay? Improve the patient care. That itself is a huge thing, okay? Yeah. Okay, from morning session, right? So we have this, is it a tea time? I didn't know. Is it? Okay. Okay.